has 17 carbon. On the 17th carbon, what is present? If there is a long chain, this long chain with a isopropyl group here, dimethyl group here, what is that? Instead of this carboxylic acid, if there were dimethyl here, what would that be? Cholesterol. Okay, so if this were instead of this, if it were this, that would have been cholesterol. So that isopropyl group has been oxidized to give you this. So is this oxidation? Converting that to this, would you say this is oxidation? Why? You're adding oxygens. That's simple, right? So you're adding. Look at this carbon here. That carbon is this carbon. Right. So you're adding the equivalent of 3 oxygens to that carbon. So this is an oxidation. So what produces this is, by the way, cholic acid. And this molecule is called cholic acid. This is one of several acids that are produced in the liver, which are collectively called bile acids or bile juices. Right? So these are bile acids. Cholic acid is one of them. So if liver is able to convert cholesterol, again, common sense, would you expect cholic acid to be produced from cholesterol? No. Common sense wise, would you expect the structure? It looks like a duck, quacks like a duck. Is it a duck? Right? It looks like cholesterol. Just 2 carbons separated and oxidized from cholesterol. So is it common sense to expect cholesterol to be oxidized to cholic acid? Yes. Which organ is producing cholic acid in the body? Liver is. So what is happening in the liver, do you think? So that's a good answer for a biologist. As a chemist, so what is the liver doing?
Oxidizing.
Oxidation. Yeah. So now for the first time in our body, are we seeing reactions other than hydrolysis?
Yes.
Right? Hydrolysis is everywhere. It's in the mouth, it's in the stomach, everywhere it's just hydrolysis. Breaking down amides to acids and amines, esters to acids and alcohols, acetals to aldehydes and alcohols. That is all hydrolysis. Beyond hydrolysis, the other key reaction that is commonly occurring in the body is oxidation. And where does oxidation take place? Liver. How does it happen? How does oxidation happen? Did we talk about oxidation 2 or 3 weeks ago in the class?
Yes.
For which context? Huh? We talked about metabolism now. In which context did we talk about oxidation? Aromatase, good. Right? In aromatase, what was happening? Methyl group on testosterone was getting oxidized to carbon dioxide. Yes? Methyl group on testosterone was getting oxidized to carbon dioxide. So how was that happening? Peroxide, but which enzyme was involved? Heme, aromatase, but what was the active side of aromatase? Heme. What is heme? What is heme? Iron. No, globule is separate. Heme is other part. What is heme? Iron followed by or connected to the porphyrin ring system, right? What is a porphyrin ring? 4 5-membered rings connected by a carbon each, correct? With nitrogens and conjugation. We talked about all that before. So that is involved. And is that causing oxidation?
Yes.
Yes. So would you expect something similar in the liver as well to cause oxidation?
Yes.
So these enzymes collectively, there are some 10,000 different varieties of such enzymes in the liver. There are 10,000 unique enzymes. There are multiple isoforms of each and so on. So essentially there are millions of enzymes. If you isolate every one of them, there will be million types of oxidizing enzymes in the liver. Collectively they are called. Collectively these oxidizing enzymes in the liver are called. These are a type of oxidizing enzymes. but they have a name. All these oxidizing enzymes in the liver are called, you might have heard of this.
Oh, CYP.
Cytochrome P450, CYP. Right? Cytochrome P450 are those 10,000 unique type of enzymes, multiple, so there are millions of subforms and isoforms of these enzymes known in the liver. So all these enzymes, are they already in place in the body?
Yes.
So humans have existed much before the medicines, correct?
Yeah.
Medicines did not come first, correct?
Yes.
So we have existed before, and all these enzymes were already in our body, correct? So the drug that we take, is that also going to experience this?
Yeah.
So when I get back to pharmaceutical chemistry eventually, we should keep all of this in mind. Right? So that's why we're discussing this so much in detail. So all of these enzymes, conditions, reactions, and so on, the drugs will be exposed to as well. Is that clear?
Yeah.
So cholesterol will be converted to cholic acid, and what happens then? The bile acids, they get added to the digested food in the stomach, and collectively they all reach which part of the body? Small intestines. Small intestines. Right? They reach the small intestines there. What is happening there? At that point, all these small molecules that are produced, like these the glucoses and then the amino acids and the fatty acids, all these type of molecules would be absorbed. Some of these fatty acids are then used in the prostaglandin synthesis. And so prostaglandins are again unique molecules that are used as hormones and such. So they are also produced and absorbed through the intestines. So all the useful stuff is going away. Is bile acid going away? Not really. It's still present. Now, again, chemically speaking, what do you think is the property of bile acid? What is the physical property of bile acid? Physical property. Physical as in lipophilic.
Oh, okay.
It is lipophilic.
Yeah.
Right? It is lipophilic. So, what type of molecules would adsorb on it? Again, is this a solid? Would you expect this to be a solid? Would you expect cholic acid to be a solid? Is cholesterol a solid? Yes, cholesterol is a solid. Cholesterol is a solid. You should have used it in bioinformatics. Right? Cholesterol is a solid. If you oxidize a large molecule to form a carboxylic acid, is it a little more polar than cholesterol?
Yeah.
Yes. So there will be even more binding interactions between molecules. It will pack even better. It will be a higher melting solid. So bile acid, it's a solid. It is a solid. Now, what type of molecules does this associate with? What type of molecules have a higher affinity to bile acids? Polar or non-polar? Non-polar. Non-polar. Why? This, yeah, like dissolves, right? So this molecule is non-polar. So other non-polar entities. So from the food that you ate, whatever is not getting metabolized, not getting absorbed and so on, what happens to it? All of that will associate itself to bile acid, assuming it is non-polar. What if your waste that is not absorbed is polar? What will happen then? It will not. What? It will not absorb. It will not absorb to this. Where would that go? It will go to the kidneys. It will reach the. It is water soluble. At that point, it will reach the bloodstream. It will be absorbed into the bloodstream, and then from there, it will do its own thing. And then eventually, all that liquid waste reaches for the kidneys. Right? And from the kidneys, they then reach the urinary tract and into the urethra, and it just goes away. Right? So that is how the liquid waste is removed. So for that to happen, what type of molecules do you think would go easily through urine? Water soluble. Right? So if something is, would you expect fat to come out of urine?
No.
Would you expect grease to what? That would be a problem. That would be a problem. Right? So do you expect solid coming out of urine?
I hope.
Usually no. Usually no.
Yeah, it is.
Yeah, but there are situations, there are conditions in which it does happen. Hyperketonuria. Some people have difficulty in digesting and metabolizing ketones, in which case they could lead to kidney stones and such. And then there are physically painful processes that could lead to that also. Barring those conditions,
Yeah.
what type of molecules are excreted through urine?
Water soluble.
So how does that happen? So for example, I have a molecule, let's say I ate oranges. Okay, I ate oranges, I have limonene in there. Okay, I have limonene in there, and most of it is absorbed, but the rest of it needs to be digested. The rest of it needs to be excreted out. So for example, when you drink coffee, a few hours later when you go, does it smell like coffee a little bit?
Yeah.
It does.
Yes.
With oranges, there are some fruits. You do some foods that do impart that smell to the urine.
Yeah,
as well, right?
asparagus, yeah.
As far as there are multiple things. So how is that happening? How is that
It's
happening?
being excreted. It's not broken down.
It's not broken down. It's being excreted. But what type of molecules are excreted through urine? Polar. Limonene certainly is not polar. Huh? Limonene is this molecule here. What? Huh? It's not through urine. Let's hope it's not through urine. All through urine would be an issue. It will be a big issue. So this is limonene. This is not water soluble.
Right.
Absolutely not. Right? This is not water soluble. Yet you do smell sometimes, right? How is this getting excreted through urine? Not like that.
Not like that.
Not like that. What does it? It should be. This is ideal to be removed from feces.
Right.
Some of it does go through feces too. Okay, some of it does go through feces. Sometimes the colors, you can associate that too.
Right.
Leafy vegetables, it gets leafy. If you eat beetroots, it becomes red.
Right.
If you eat too many grapes, that color comes up. Right? So it's all associated. So non-polar stuff, that's the easy part. But through urine, how is that going out? A molecule such as this, how would this go into the urine? It has to become more polar.
Right.
Okay, it has to become more polar. How does it become more polar? Or how do you make molecules more polar? Good. You can add an alcohol group. Would it make it a little more polar?
Yeah.
Yes. How about an amine?
Yes.
Yes, it will become more polar. To make an ammonium salt, and more polar.
Yeah.
Right. Now, how do you add an oxygen or an amine to a molecule? What is that process called? Adding oxygen is called oxidation. Just a few minutes ago, did we talk about oxidation?
Yes.
Where is it happening? Okay, what? Are there CYP enzymes? So CYP enzymes are able to oxidize molecules, and they'll add, say, to this molecule. Could you foresee, say, for some other oxidizing enzyme, adding an OH group somewhere else?
Yeah.
Could that alcohol be further oxidized to a carboxylic acid? Sure. Right? Could you add multiple hydroxyls? Sure. Could you add an amine? Sure. There are multiple hydroxylating and aminating enzymes in the body which could oxidize. But there are 10 odd carbons in this. If there are 10 odd carbons in this molecule, adding an extra one OH group or two OH groups or an amine group. Yes, it increases the polarity, but does it increase it so much that it will be freely soluble in water? Because again you have to remember, if salt versus sugar, there are differences in solubility with water, right?
Yeah.
A lot of salt can go into water, but only a little of sugar can go into water. Why? Polarities are different. Right? Polarities of sodium chloride and sugar are different. So that tells you how much of a molecule can go into water. So adding an alcohol to this will make it somewhat soluble, not extremely. So let's say you have a few milligrams of this in the urine. How much water might be needed? You might have it, let's say, a milligram per liter, let's say. A milligram per liter. So you have to pee out of a few gallons to get it off this. So you have, that means you have to drink that much water. That's ineffective. Yes? Just adding an extra oil, and limonene is a small molecule. It's a fairly small molecule. There are much bigger molecules present in the foods. So then how do you get rid of those? Adding one OH group or two OH groups will not help, will not be enough to make it freely soluble in water so that it can be excreted readily. What do you do next? It's not possible all the time. So that is a possibility, but it's not possible all the time. Especially again, if we keep our drugs in mind. With the drugs, the processes are not in place to convert those to small molecules. So what else do you need to do to make it freely soluble, so that large amounts of this molecule can go out in water? Very good, very good, very good. Right? So if a molecule is already, if the molecule is not polar, you can add some groups like OH, NH2, SH, and so on to make them somewhat more polar. And then to this polar molecule, you add an even more polar molecule. What is an even more polar molecule? Sugar.
Sugar. Yeah.
Is sugar polar? Yes. So what is sugar? What is the simplest sugar? Glucose. What's the structure of glucose? Right? That's glucose. Is that right?
Yeah.
So 6 carbons, an aldehyde, and a few alcohols. Or you can draw it in that fashion as well. That's the cyclic form of glucose, glucopyranose. Is this molecule polar? Why? There are 6 carbons and 6 oxygens as well. There are 6 carbons, and for every carbon you have an oxygen. It's a highly polar molecule. So does this molecule dissolve much more freely in water than, let's say, this or its oxidized form? Absolutely. So if this molecule is polar, what would you say this molecule is? Even more polar?
Yeah.
Right? Glucose is an aldehyde. Right? There are multiple alcohols and an aldehyde. Is an acid more polar than an aldehyde? Yes. So would this molecule be even more polar than glucose?
Yes.
And the advantage of this molecule is now you have an acid. If you introduce an alcohol or an amine here, could you couple these two?
Oh.
Right? So this is now an acid. If you introduce an alcohol here, could you couple this acid with this alcohol?
Yes.
What does it form? An ester. Introduce an amine here, couple it with the acid, what do you form? An amide. Right? So is there an easy linkage between these two molecules now?
Yes.
So are you facilitating the process, are you now enabling the non-polar molecules to become exceptionally polar and freely dissolved in water so that you can remove the urine? So this is a two-step process. In the first step, this molecule, can this bind to this molecule?
No.
Not really. So there has to be a preparation. You have to prep this molecule to tag with this molecule. So the first step would be referred as a simple oxidation. That would be phase 1 oxidation. Where does that happen? That would happen in the liver.
Oh,
That could happen in the liver. These CYP enzymes, there are multiple oxidizing enzymes available. They could technically oxidize this to some alcohol, some amine, and so on. And in the second phase of metabolism, what would then happen? That oxidized product would react with this molecule. What is this? This is glucose is aldehyde. Right, so this without the OA is glucose. This is this glucose acid? Is this glucose acid? So it's called glucuronic acid. Okay, this is called glucuronic acid. Okay, this molecule is called glucuronic acid. So when you add a glucuronic acid to an alcohol, what would you call that process? Glucuronidation. Right? So this glucuronic acid, when it is coupled with an acid as an alcohol or an amine, that process would be called glucuronidation. So next time you read some metabolism processes, it will make more chemical sense now. Right? So this is the chemical process happening behind metabolism. So is this how you get rid of molecules via urine?
Yeah.
So this is excretion. Now, I want to talk about defecation first. But so going back to defecation, what's happening now? So your molecules have been chopped down to small pieces. Whatever is absorbed through water and such go into the bloodstream, and then they go do their own thing, and then eventually that waste ends up in the kidneys after it has been oxidized and glucuronidized, and then from the kidneys they go to urinary tract, and then they're eliminated. Yes. That's one process. So the next step would be, the other thing would be, whatever is not possible to oxidize, what will happen to those? Something is non-polar. It doesn't dissolve in water. It cannot be oxidized in the liver either. What's happened then? That stays put. That stays put. And would that now see cholic acid and other bile acids?
Okay.
So that the non-polar molecules that cannot be oxidized, they cannot be glucuronidized, they will adsorb. And they'll adsorb onto these bile acids, and they are now in the intestines. Right? They're in the intestines, and even from there, if there is anything useful to be had, so you understand that the small intestines are like meter long, right? They're really long intestines. Again, herbivores have an even longer intestine, small intestine, than carnivores. That is a fact. Cows, buffaloes,
Oh, yeah, no.
cows, buffaloes, when compared to tigers and lions. Okay? Herbivores versus carnivores. Herbivores have, when dad explains, but that's how I explain my and I excuse. Okay, so herbivores have longer small intestine, and more than the length, what is the actual shape of the intestine inside? It's even within its curve.
Yeah.
Not only is it long, it's curved within as well. What does that lead to? No, not the fact. It increases
Surface area.
surface area.
Oh, okay.
It increases surface area. What is that? Why is that critical? Absorption.
Yeah.
You're exposing the molecules to a greater surface area as it is moving through the distance. Right? So much chemistry involved there, right?
Yeah.
So chemistry, physical properties, absorption, all those things that we learned back in chemical chemistry, they're all relevant here. So when all this is happening, everything that is useful to be had will be absorbed through the small intestines into the bloodstream, and then even after this arduous process. Whatever doesn't get through, whatever is not absorbed, what will happen to that? That will reach the colon, larger distance. Right? It will reach the larger distance. Larger distance is relatively small. It's not as long as the small distance. What is happening there? There is some amount of absorption going on in the larger distance, not much.
Yeah.
What is the singular process there? It sucks out the water from there. Okay, so any remnant water is removed. So as a result, now you have all this waste. It's getting all the important ingredients are absorbed. Whatever is left out is still loose. It goes to larger distance. That's where all the water is being sucked out as well. So what are you left with? A solid mass. A solid mass is left out, and then that then travels about I think about 5-6 hours is what it takes to go through the intestines as well, and another 3-4 hours is the time that it spends in the colon, unless there are issues. So then it goes through the colon, and by the end of it, it then reaches the rectum. It reaches the rectum, and that's how you inject it out. Right? So that is defecation 101. Good? Okay.
How would fiber affect this? How would fiber like take the swing on affect this process?
Good question. How does fiber help it? How does taking fiber help it? Fiber is what? Fiber is essentially solid particles that are not easily digested. It's chitin and other. These are the almost like the plant tree cellulose, but chitin cellulose. Chitin is a specific form of cellulose. It has some aromatic rings in it also. Chitin has aromatic rings connected to cellulose and such. So that's why, for example, the trees, we cannot eat tree trunks, right? Can eat potato, but not tree trunks. Buffalo can eat tree trunks too, because of the long distance. It has longer time. Again, we should not get into that. Let's stick to humans. So fiber, because of these highly non-reactive molecules that cannot be easily oxidized, that cannot be easily digested, yet are solids. So what does that help with? Is it now providing a solid surface on which all this waste can absorb? Does that help with forming a better solid lump?
Yeah.
Not all of it though. So it's not just, you're not eating the tree trunk directly, right? Hopefully not. I mean, if you don't chew and swallow the whole thing, then maybe yes. Okay, good?
Yeah.
So that's what the fiber is. Fine. Let's now get back. I was not planning on spending this much time. Let's get back to the pharmaceutical chemistry. Okay, so we now understand our body at least a little bit, right?
Yes.
Okay. So now we're taking medicine. Okay. What are the various forms in which you can take the medicine?
Tablet, enteral fluid, intravenous fluid,
Intramuscular, subcutaneous,
Ocular,
nasal.
basal,
the rectal,
Yeah.
the rectal tube,
Vagina.
the rectal tube, right? So, for example, for infants, who don't take Tylenol and such, the medicines that don't, if they throw up, what do? You inject it. You have small pouches that can be inserted rectally. That is also, so there are multiple holes through which you can put stuff into our body. Right? There are 9 holes. All 9 holes are fair target. 9 holes, and that's what I said. 9 holes. Okay.
10 sometimes.
So you should be able to inject it any which way. Most common ones being oral and injection. These are the most common ones. Patches and other things are always there. So let's talk about the oral one. That's the most obvious one. The most common one is oral, correct?
Yes.
So, is that drug being subjected to the exact same situation as our food?
Oh,
Yes. Yeah. Yeah. So then, the drug that you're taking, what does it need to withstand for it to be able to be a drug?
pH.
pH 2. If your molecule dissolves or decomposes at pH 2, is that molecule unfit for oral delivery?
Yes.
Then your only option that becomes injection or something. So that's called the first pass metabolism. In the first pass metabolism, it has to withstand it, otherwise that drug will never make it. What? What if it is made? That's fine. Then it's okay. That's okay. Okay. So the acidic environment it has to withstand, and then it has to withstand the basic environment in the intestines as well, and be subjected to the liver. Right. So the liver has what type of enzymes? Step enzymes, cytochrome enzymes. Are they oxidizing enzymes?
Yes.
So are there a lot of reaction possibilities for the molecules then?
Yes.
So how does a drug get metabolized then? What are the main reactions that you need to associate your molecules, your drugs with? Now only two reactions talked about so far, right?
Yeah.
You know, all the defecation, excretion, all these processes, only two reactions came. They are redox, oxidation reduction, or hydrolysis. So if your molecule is not prone to either, what will happen? If your drug that you're synthesizing is not prone to hydrolysis or to oxidation, what would happen to it? Let's say it's 0% hydrolyzed, it's 0% oxidized, what would happen? It will either accumulate in the body if it is going to be absorbed into the fat or whatever, or it will simply be 100% excreted out. So would that be effective
Yeah.
as a drug? Not really. Right? So if it is not excreted, it's not getting metabolized, right?
Yeah.
So is that a problem?
Yes.
So this was a long-winded answer of my original question at the beginning, right? My original question, which led me to this path, is why would you need your molecule to be readily metabolized or not? Or how would a drug get metabolized? Now you know. How does a drug get metabolized? Oxidation, reduction, and hydrolysis. These are the three main reactions you need to contend with to make a drug more metabolized or less metabolized. So, are either or both useful
Yes.
depending on context? How? So let's say somebody is suffering from high blood pressure. They take a medicine and it takes 6 hours to take effect. They could just die before that, right?
Yeah.
The blood pressure shoots up really high. And what could be a reason the drug is acting so slow? It's not getting metabolized, it's not binding to anything. It's a good drug once it gets metabolized, but it takes 6 hours instead to metabolize. So is that a problem?
Yes.
So how would you make that drug effective? You make it more metabolized. So the technical word for this is you would want to increase its metabolic vulnerability. Right? You would want to make this drug more vulnerable to metabolism. Vulnerable means more prone to metabolism. Right? So you would want to increase the metabolic vulnerability. Now consider another situation. Somebody suffering from diabetes or we can say diabetes, and you take a medicine, it goes out of your bloodstream in half an hour. Okay, half an hour, 10 minutes, say the whole molecule is getting integrated. In 10 minutes you pay out the whole drug really fast. Yeah. So that's also bad.
Yes.
Right? Under those situations, would you want the drug to be long lasting?
Yes.
Yes. You would want to decrease, So why is it getting rid easily? Because it is getting metabolized faster. So would you want to reduce the metabolic vulnerability?
Yes.
Example, there's a schizophrenia patient. You have to administer a drug. The drug takes a day for it to last. Its effects last till you take it. The next day, somebody has to administer because the patient may or may not remember whether they took it or not, or they may be non-compliant. So what do? And then some of these drugs could be so nasty that they cannot be sold. They cannot be just sold in retail and such. These have highly regulated drugs, where if the illicit parties get hold of these drugs, they could make contraband, they could make other illegal drugs. There are multiple problems associated. So if such a situation exists, what is a better thing to do? Administer it at the facility, right? In the doctor's office, in the hospital, and such. But if the patient has to go in every day, that causes compliance issues too. So as opposed to now, let's say if a drug is able to last a month, now is it much better?
Yes.
Coming into the facility once a month, get an injection, go. So what do you do then? Increase or decrease the vulnerability? Decrease. So increasing or decreasing the metabolic vulnerability would be a good thing depending on the context. How do you do this then? How do you make a molecule more vulnerable?
Make it more likely to be oxidized
Or reduced, right? So make a molecule more likely to be oxidized. How do you make something more oxidized? More readily oxidized? What's it? Not every polar molecule is oxidized. Polar is not the word. What would you want to introduce? So how would you make something more readily oxidized? Good. You could make it more electrophilic. Right? So if you increase the positive character on a carbon or on a hydrogen, that is more prone to oxidation. So something like benzylic acid as benzene versus toluene. Benzene versus toluene. What do you think is more prone to oxidation? Benzene versus toluene. What is more prone to oxidation? Toluene is because the CH3 group, this CH group, that's partially positive. That can be oxidized to a benzylic alcohol, benzaldehyde, benzoic acid, and such. Benzene though is not prone to oxidation as much. So I have a drug which has a benzene ring on it. It's getting forever to oxidize and metabolize. What can you do to increase the reactivity? Put another group on there. Put an alcohol group on there. Put a CH2OH group on there. So put some type of groups that can be readily oxidized. Then you are increasing the vulnerability. And how do you achieve the reverse? So you have a drug, it's getting metabolized too fast. You find out that there's a methyl group on a benzene ring, or there's this primary alcohol group on a molecule. What do? Or get rid of that group. You have an alcohol group, get rid of it. Make it ether. Ether is not oxidized easily. So convert alcohol to ether. Or you have a benzene ring or methyl group. Get rid of the benzylic hydrogens. Instead of methyl, let's say T group. So there are chemical ways of figuring out how to affect the reactivity of a molecule under the biological conditions. For that, now the really long-winded answer is molecular modification critical. Is that how you change the properties of a molecule?
Yes.
That is where your design comes in, right? So the question is, in this modification, what if you end up changing the pharmacophore? Then that just defeats the whole purpose, right?
What if the only area is to
then that's not a target. That's not a good target for molecular modifications. You find out other avenues of making it better, not using molecular modifications. Okay. So the question is, if the only location possible on a drug is the pharmacophore that you can change, and to effect the property that you want, then you have no choice. You just cannot do it. You just move on to the next drug, or you do something else to make this drug effective. Okay, molecular modification is not the route for that. You would want to make maybe a different type of formulation, maybe a different type of administration, maybe make the drug into a nano formulation, a nano form instead of the macromolecular structure. Find out other ways of achieving the properties, not via molecular modifications. Is that clear?
Yes.
Let's consider the situation. I have a drug. It's not as soluble in water. As a result, it has to be injected. Okay, let's say it has to be injected. It's not orally available. It has to be injected. It is not soluble in water freely. So what does that mean? I have to say, if I have to give a 5 milligram injection, for 5 milligrams, I may have to use 5 milliliters of water. So your injection size becomes bigger, right? That could be detrimental, right? Or if the injection comes in the form of a liquid, if the solubility is sparing, upon storage, solid might crash out. Yes? If the compound is not freely soluble in the medium in which this injection is made, it will crash out over time if you leave it in the fridge, because that's where you're typically storing your injections, right? So they could crash out. There are physical challenges associated. So how do you address that? The drug is good. I don't want to change the drug. I want to put in millions of dollars investment into that drug to make it effective. So I don't want to kill that. So what do I do? I would want to now make it more polar. More polar. How do I make it more polar? I can learn from nature. Right? I can learn from nature. What did nature do? Add a polar group to make it more polar. I mean, I'll not go all the way to glucose, because that's going to be so soluble that you just directly pee out of it. Right? That's not useful either. So I would want to moderate this solute. So how do I do this then? I'll find an alcohol group, I'll find an amine group, I'll find an acid group on my molecule, on my drug, and try to put in some polar groups on it. Examples of polar groups would be glycerol, citric acid, these type of molecules have several polar groups on them. So these type of polar acids, would I be able to couple them with the alcohols or amines on my drug and make a more polar molecule? This would be an example of a molecular modification too. Yes?
Yeah.
The reverse. The drug is too soluble. It's really good, but it's so soluble that 80%, 90% of it is just unchanged, excreted through urine. Okay, so you have a drug that is effective, highly effective, yet 80-90% of the drug is not being used in the body because it's simply being excreted through urine, because it is so soluble in water. What do you need to do there? Make it non-polar. You would want to make it non-polar. How do you make it non-polar? Instead of something like alcohol, polyalcohols or polycarboxylic acids, you would want to put something like oleic acid, palmitic acid, stearic acid, some 16, 17, 18, 20 carbon chains, acids, and so on. And that is how you can increase the lipophilicity and reduce the water solubility. You can reduce the. If you're not impacting the pharmacophore, right? If you're not impacting the pharmacophore, you would get away with. And the key there is to make sure whatever group you're putting on, there are some conditions too. So you cannot just put random things on there, right? Let's say this group that you're adding is an extremely potent toxin. Would you want to put that on your drug? No, because that will get released in the body. It's a closed system that we have, right? Except for those 9 holes, it is a closed system. Right? And it just automatically doesn't go to those holes. So everything that we're taking in is still inside us. So if any of these have toxic side effects, you have to bar those from these manipulations. And that's not all. The molecule itself may not be toxic, but once it enters the body, upon oxidation or upon some modified metabolism, the metabolite. What is the metabolite? Metabolite is the byproduct, is a product of metabolism. So we talked about this, for example, MTX. Did we say MTX is an anti-metabolite? What is an anti-metabolite? Something that prevents the formation of metabolites. THF, for example, is a metabolite. THF is a metabolite of folic acid. MTX prevents the formation of that metabolite, so MTX would be considered an anti-metabolite. So not only do you have to contend with the toxicity of the groups that you are putting in, you also have to worry about how these groups are getting converted in the body and what are the properties of those groups as well. So your choice is fairly limited, right? Your choice is fairly limited because if you get too exploratory with these groups that you're putting in, you do not know what the long-term repercussions of these new groups that you're adding are. Yes? So you would not want to be exploratory here. You would want to be pretty conservative. Use the existing things. For example, succinic acid. Is the body already adept at using succinic acid? Yes. Tartaric acid, maleic acid, fumaric acid. Right? All these are already existing drugs, existing molecules in the body. So use those. Citric acid. All these are naturally occurring molecules. So use those as targets, rather than something random, something new. So benzoic acid, you would not want to use benzoic acid because that's not a normal existing molecule. Are we good? So we talked about that. Taste. If your drug is extremely bitter, and it's a children's medication, what do you do? What's it? On. Yes, so there are chemical ways of doing it, and there are non-chemical ways of doing it, and there are forced ways of doing it. So you can just force the kids to do it, they take it, but it doesn't work all the time. They'll just puke it out.
Yeah, that's
Okay, so that's one possibility. Or physically, what do you do? What can you do? If the drug has a bad taste, you can add some syrup.
Yeah.
You can add some sweetener or flavoring agent or some tasting sweetening agent. That is a possibility. But chemists, as scientists, we cannot just let go at that stage. That's more of a formulation, not a chemistry job. So what can you do then? You can make changes to the structure. You can modify the structure in such a way that the taste is altered. How do you alter the taste? That's a risky answer.
Yeah.
No, forget the polarity. That's a very risky answer because just because glucose is sweet does not automatically mean that every conjugate of glucose is sweet as well.
Right. Yeah.
Right? So the moment you introduce something else onto glucose, it is a new molecule. There is no guarantee that it will be sweet. Okay, so taste, properties you can manage. Polar, non-polar, that you can manage, that you can predict. Taste you cannot. So by default, saying you'll couple it with glucose is not going to cut it.
Are there actual like molecules or like groups that we can add onto the
taste is a fairly less understood. It's a highly less understood field in the body. Taste is very less is known about it.
You could add a molecule that has a higher affinity for a different receptor over that particular taste receptor. Like if it's a bitter receptor, you could have something that maybe blocks that or preferentially.
That level of detail you can do, yes. An even easier thing. What do you taste on your tongue? You taste oil.
Yeah.
Oh. Is there taste of oil for oil other than olive oil? For olive oil. Usually do oils have taste?
No.
No. Does the salt have taste?
Yes.
Yes. Sugar have taste?
Yes.
Yes. Bitter things. So what do all the tastes have in common? They're water soluble.
Yeah.
They're water soluble. At least somewhat soluble in water. And our tongue, that mouth is all water, hydrophilic, water aqueous medium. So if a molecule doesn't dissolve in water, or if it has lesser solubility in water, is it highly likely that the taste will change?
Yes.
Right? So there are subtle ways like that which you can do as well. Again, I'm not saying it will work every single time for every single drug. That's right.
No, you said, sorry, you said again, you said taste can change based on what?
Taste can change based on how polar or non-polar the molecule is. Because if the molecule doesn't dissolve in water, is that the equivalent of oil?
Yes.
Right? So why don't you taste oil? Because it doesn't dissolve in the tongue. Most of your taste buds are, or all of your taste buds are on the tongue. Correct? All the taste buds are on the tongue, and it is aqueous. If something doesn't dissolve in water, then it's it's less likely for you to be able to taste it. Okay. So olive oil has a taste because it has a slightly polar molecule called oleocanthal. There is a molecule called oleocanthal which is an aldehyde. It is somewhat polar, so that's what you taste in olive oil. Are we good?
Yeah.
So there are various reasons why you would want to make modifications to an existing structure, existing drug. Yeah. That is a possibility too. That is a possibility as well. But that's a formulation, that's not a chemical. Which is fine, but as organic chemists, do we accept defeat like that? No.
Wouldn't it be cheaper to go that way than have like syrups and like,
Sure. Sure. But isn't it also easier to do it? Is it easier to do it? Yes. So if you can do it, so can 100 other companies around you. And if the drug is the same, it's simply a generic. You make a formulation, I make a slightly better formulation. So you're out of business. Right? Yes? What's it?
No,
But again, that's like the cynical reason. But as chemists, as organic chemists, we cannot accept defeat like that. And so we have to have an answer for that. Okay. Talked on this slide for an hour and a half. What's it? Okay, so there's a lot of text. Start reading it. I think I've said everything here that I need to say.
Yeah. Pretty much what we.
Okay. Look at the. What's that?
So this is how we do our presentation at the end. This is how we do our presentation at the end.
So look at the molecule carbutamide. Okay, carbutamide is a diabetes drug. Okay, carbutamide is a diabetes drug. But it has antibacterial side effect. Is it a side effect? Antibacterial, is it a side effect?
Yeah.
As I may have told you before, one of my friends used to say, when you're trying to look for silver, gold is an impurity.
Yeah.
I may have said this before.
Yeah,
So
yeah.
if your goal is to extract silver from the ore, gold, even though it is more precious, is still enemy. So if your goal is to make a diabetes drug, having antibacterial properties is not what you want.
Right.
So that is a side effect. Yes?
Yeah.
Now, all that aside, why is this a side effect?
What?
Having an antibacterial
Because it doesn't discriminate in your microbiome, so you can kill the good bacteria.
Absolutely. Our body has a lot of symbiotic bacteria too, right?
Yeah.
Beneficial bacteria. So an antibacterial side effect would kill all those good bacteria as well. That could cause digestive problems and other issues because some of these bacteria have a symbiotic relationship as well. So a lot of other properties, properties in the body, processes in the body can be affected as well, right? So, is there anything about carbutamide that tells you that it should or it could have antibacterial side effects? Is there anything about carbutamide that gives out a clue that it should have antibacterial side effects? Good. Good. The NH2 connected to the benzene ring, connected to a carboxylic acid, was what molecule? NH2 connected to benzene ring, connected to carboxylic acid. What was that molecule? Was that what we started with today, in the beginning?
Yeah.
What was that molecule? Para, para-aminobenzoic acid.
Yeah.
And what was the drug that we discussed based on para? What was the drug that we discussed based on para? Not folic acid. What was the difference between bacteria and humans? Yes, the ability to synthesize folic acid versus not. So we cannot, bacteria can. How does that happen? How does the bacterial folic acid synthesis happen? What is the key intermediate? What is the key intermediate involved in the synthesis of folic acid within bacteria? What's it? That's the precursor. That's a precursor. What is it reacting with? What is stearin diphosphate reacting with? Look it up. What is stearin diphosphate reacting with to form folic acid in bacteria? Which acid? It's an acid with a benzene ring and an amine group. A carboxylic acid on a benzene ring with an NH2 group on the fourth position. Amino-benzoic acid. Para-amino-benzoic acid was reacting, correct?
Right.
If you remember the beginning of the semester, we had talked about how folic acid is produced in the bacteria by reacting pterin diphosphate with para-amino-benzoic acid. So what is the structure of para-amino-benzoic acid? Right? That is called carbon. And from there, what did we do? How did we discover a drug for this? We said bacteria consume this as food through their intake, and I use this to produce folic acid. So what did we do? We changed. NH2 was critical for it to react, correct?
Oh, yeah,
Oh,
like
okay.
that's the
The OH, the NH and the OG.
No. What did we change?
Carboxylic acid got used like a kind of like a sulfate.
Yes, sulfonic acid. So this got changed to sulfonamide. That was way at the beginning. Okay, probably like the third or fourth class of the semester, which talked about this. What? Third or fourth class in the semester is when we probably beginning of February. Beginning of February is when we talk about this. So para-aminobenzoic acid. We replaced carboxylic acid with sulfonamide. Because sulfonamide and carboxylic acid have similar pKs as well. And this NH2 still reacts. So is this a part of sulfur drug? This is a part of sulfur drug. Now look at carbutamide. Right? Does it have up until this sulfur? And in fact, between this nitrogen. Right? Up until this structure is it the same as carbutamide. So this has diabetic side properties, but is it a surprise that this is antibacterial as well? Would bacteria eat this? Absolutely. And what would happen as a result? It will prevent the folic acid synthesis. So are you surprised that this has antibacterial side effects? What's that? No. So what do? It's a drug. It's a drug. And what you also learned is the NH2 is not a pharmacophore. What you also learned, how do you learn this? You just make random molecules, get rid of the NH2 group, and still see if it has antidiabetic side effects or not. They found out that it is not. So what do you need to do? What should you do? Get rid of the NH2 group. And get rid of the NH2 group, and what do you put instead? Anything. Anything. Sky's the limit. Just put some random group on there. Check the properties. Now, at what stage are you at the drug discovery process now? You have carbutamide, it is an existing drug. Over a period of time, you have the patients are reporting antibacterial side effects. So, as a chemist, your job is to make it better. So you came up with some new drug. Let's say you make carbutamide. At what stage in the drug discovery process do you go back to? What are the various stages of drug discovery?
Structural activities.
Structure activity followed by? Give me brief chronological bullet points. Identifying a target, structure activity, cell studies, enzymatic studies, then animal studies, IND, followed by phase 1, phase 2, phase 3,
Phase 4.
NDA, and then approval. Approval followed by phase 4. So carbutamide is already done. It has passed through all these stages and is now in the market. Right? When does the molecule get to the market? After all these stages are done, right? So now the question is, after all this, there are side effects. So still, some people may still use it, but over time, it will just become not practical. So the company, what does the company do? It would want to invest in R&D. It would want to make newer molecules. Let's say they came up with another molecule that is also effective, carbutamide, and they also found it that doesn't have side effects or antibacterial side effects. So now my question is, at what stage in this drug discovery process that we discussed a minute ago do you start? So let's say carbidopa is already a drug and change the NH2 to CH3. Can I just give it humans now? What's that? You have to apply for another IND.
Yeah, right.
How do you apply for an IND?
You have to show the animal and
You need to show the animal studies.
yeah.
So to do the animal studies, you need to know what dose to give and such, right?
Yeah.
So where do you start? You go back to the beginning. Right? The only thing that you skipped was all this elaborate SAR, and the only thing that you skipped was SAR. Even then, it's not, you didn't technically skip it, because all I'm showing here is the final product.
Yeah.
Do you think this is the first molecule they tried? Maybe, maybe not. Right? There's a good chance the first molecule works, or this might be the 10th iteration. So that is trial and error, the judicial choice. All those are still part of this. So SCR is not technically out. So you have made one simple change. Changed an NH2 to CS3. Which part of the drug discovery did you go back to? To the beginning. Right? So how much would it cost?
Billions.
Probably the same thing.
Yeah.
But would the company be much more confident of this drug possibly working than a random molecule that
Yeah.
there's a high chance, there's a very high chance this will actually work, because similar thing, almost the same thing is already in the market. So long as they're able to have a relatively non-toxic molecule which is efficacious, there is a very high chance that tolbutamide will become a drug. So from a business standpoint, would the company, would the CEO at the board be less vulnerable to taking this up? So would they be more likely to take up this molecule over some random molecule?
Oh,
Yes.
Does it make sense? Even though it is going to cost this much, they have a higher chance of success with this molecule than a random molecule. So would you consider tolbutamide a second generation drug?
Yeah.
Would you expect this to have antibacterial side effects? Tolbutamide. Would you expect tolbutamide to have antibacterial side effects? Not really, because that NH2 group that was responsible to bind to the terrin is no longer there. Yes? So is this an example of a molecular modification? So chlorothiazide is a blood pressure medication, antihypertensive drug. It is a diuretic as well. Okay, what is a diuretic? It increases the urine production. Okay, so diuretic, coffee for example, is a diuretic. Okay, so a diuretic is a molecule that is a compound that causes increased urine production. Why is that bad? Dehydration. Right, so that is increased urine production is also a condition. So is that a side effect now? Absolutely. And some commonly existing, so at this point, what do you figure out then? This drug is used as a blood pressure medication, as a high pressure drug. It has diuretic effects. What do you do then? So you have the company, you have the pharmaceutical company that developed chlorothiazide, okay, for hypertension. And now you're seeing in patients that there are diuretic side effects. What do you do as a company then? Because how do you address it? How do you make the modification? Where do you modify? You need to first figure out what is causing the diuretic side effects. Right? You have to figure out what is causing the diuretic effects. How do you figure that out? Yeah.
Just start disrupting certain structures.
Sure, that will be the second step. So making random changes takes time, takes energy, takes money. Before you can.
Or you look at other common diuretics and see what's in.
Yeah. So before you even
Okay.
before you even get to modifications of structures, see what is out there.
Right.
Let's see what is out there.
Yeah, that makes sense.
Right? So what is known is that some of the common diuretic drugs, diuretics is a condition too. So increased urine production is a therapeutic goal as well in some patients. So some of the common diuretic drugs, what do they have in common? What do the three drugs at the bottom? They all have a chlorine. They all have a sulfonamide, orthosulfonamide, connected to a benzene, right? All those are common. Just look at the commonalities. What is common? Ballpark commonality in all three. Orthochlorobenzene sulfonamide. Yes? Orthochlorobenzene sulfonamide is common in all of them. What does that mean? Ortho is 1, 2.
Yeah, one, two.
That's 2. 1, 2 positions are called ortho. So orthochlorobenzene sulfonamide. Okay, this is common amongst all these three drugs. And now if you look at chlorothiazide, does it also have the same orthochlorobenzene sulfonamide moiety? Right? So chlorothiazide. So this is how you use common sense. Okay, common sense, chemical intuition, and logic to figure out why the side effects are. Because this is easier to do, right, than actually making the molecules and purifying and testing. Just first go to the literature, see what's out there, and then compare, and then start realizing. It's not easy. Okay? In this case, it was easy to find out, but it's not always obviously apparent. But first step should be this: see what is out there. Can you explain any of this? And so the moment they figured out, so again there are thousands of drugs out there, only a few made it to this list, right? Because there are very few examples like this. But whatever is a success story is what I'm teaching you. So once they figured out that orthochlorobenzene sulfonamide is the culprit for being a diuretic, what do you need to do? Get rid of it. And get rid of it. And then they also realized that the sulfonamide on the other side, the pyridyl sulfonamide structure that you see on the other side, on the other ring, that is a pharmacophore. So they had to keep that on there. How do you figure that out? They would have figured out in the beginning itself when the molecule was being synthesized, because they would have made changes every part of the molecule. They would have realized that that side cannot be messed with. So they kept the right side ring intact. The left ring is where they are changing. And then how many changes can you make? The millions. Unlimited changes you can make. How do you figure out what is the correct combination? How do you know? So of the millions of the choices, what do you make? Of the unlimited possibilities that you have, what do you make?
Whatever's cheap to buy, and
That's one way of doing it. That is one way of doing
whatever's
it.
commercially available.
Whatever is commercially available, whatever is easy to produce, whatever is cheaper to produce, all those are good answers. But logically, what would you do? And that's what you would do in the lab. I don't teach that in class, right? What can we teach in class? I mean, practically, yes. What you said is absolutely true. That is what might be happening practically, but nobody will admit that in public.
Would this be another like structure relationship or?
That's what the question is, right? How do you decide what structures to make? The structure activity relationship is established after the synthesis is done and biology testing is done. Right? But of the molecules, how do you make them?
Literature.
you understand.
Yes.
This is
Can
the.
I hear the question prompt one more time?
So the question is, we know chlorothiazide works. We know the pyridyl sulfonamide moiety, the ring B or the ring on the right, that is the pharmacophore. You do not want to mess with that. You also know that orthochlorobenzene sulfonamide, this ring on the left, right? The ring on the left of chlorothiazide, that is the culprit. That is the reason why it has a diuretic side effect. So now the question is, of the unlimited changes that you could have made on the left ring, which change would you start with?
Just take them off.
What?
You just take something that's already present up, so like they took the sulfonamide off, and then obviously if they can work, and then, I don't know, take stuff that's present in other moieties off so that it looks different.
But again, logic has to be there, so you cannot just say take out the molecules. You should have an answer.
So, can you repeat the question one more time?
Chlorothiazide is a drug. The ring on the right is the pharmacophore. Ring on the left is the reason why it has the diuretic side effects. So your goal is to make changes to this drug so that it doesn't have those side effects, the diuretic side effects. So of the unlimited changes that could happen in that molecule, how do you decide which changes are the most effective or prudent to do? Computation. Computation would be, in this case, the best answer would be computation. I'm not saying it will work, but that is where we would start. How do you start? So the hypertensive active action, it's most likely acting on a receptor, on an enzyme, on an ion channel. There is some mechanism established already for that, right?
Right.
Before it became a drug, they would have figured out that mechanism. So using that mechanism, you see how where chlorothiazide is binding. So there is, let's say this is adrenergic receptor agonist or something like that, say, or a beta channel blocker. It's either an antagonist or an agonist of the receptor or some inhibitor of the enzyme. There is some mechanism through which it is doing its job as an hypertensive agent. You then look at the crystal structure of that receptor, that enzyme, that ion channel, whatever, and then dock it on the computer with chlorothiazide, and then just make random changes. So this is where what you said, Chris, or what you said, Lucas, just put random things on there. We're not actually making them. Right? We're not actually making them. Just put random changes on there. Put it in the system, dock it, figure out what causes the most effective binding. Simulation, computation. So on the computer, so for example, Dr. Wu, that's what his specialty is.
Yeah.
These are docking studies. These are called docking studies. So once you understand, once you know what the mechanism of a drug is, you should be able to take that protein, that receptor, that enzyme on a computer. The crystal structure would be known, and on that structure, you would insert your drug of choice and see how effectively your new molecule is going to bind. So, and you let's say do a million molecule study. In the lab, if you make those million molecules, it will take you ages. On a computer, it only takes as long as your computation time to draw the structures. And in the day of AI, it's even better now.
Yeah.
Right? You should be able to write a program in AI, have it generate those molecules, and then automatically compute it as well in a supercomputing facility, clusters. The computational speeds have increased quite tremendously now. So how does this help? Of the millions of molecules now, let's say you pick the top 5, top 10, and then you make those. Right? And then test them. So this is how you can be judicious in the type of molecules you're making. If you're starting from scratch, that's different. Then you're throwing in the dark. But now, chlorothiazide is already a drug. The mechanism is very well established. So under these conditions, it should be relatively straightforward to come up with newer design, newer analogs using computation techniques. and then you find out, let's say 5, 10 molecules, with the top 10 molecules. Then how do you decide between those 10 what do you make? That's when the logic comes. That's when your answer. Whatever is the easiest to make, whatever is cheaper to make, whatever is faster to make, whatever is readily available. You use those criteria to determine what molecule should be made first. Is it clear? And so from that, they found out that diazoxide simply removes the sulfonamide and swaps the chlorine. That proved to be the most effective, and that became. So maybe it happened in the first iteration itself, or maybe it happened in the 100th iteration. So like what you said, probably they started with the chlorine in the exact same location, they got rid of sulfonamide. And when they were making it, they saw that the metachloro, parachloro, octachloro, all those are commercially available. They probably tried all of them. Most likely, I'm guessing, the first iteration itself, they were able to figure it out. But I could be wrong. The other clues that you can follow is potency, for example. This is not a universal thing. These are now more like empirical facts. What does that mean? In general, this is the expectation. Okay, that is what this is implying. I'm not saying this is a universal fact. Okay, this is not a universal fact. This is mostly correct. This is mostly observed. What does that mean? So what is that? So one of the factors that you know is primary amines. Primary amines are in general more potent than secondary amines. Secondary amines are more potent than tertiary amines. That's a very bold statement to make. It's a bold statement to make. But can you come up with some rhyme and reason to this? What would make something more potent? When something readily reacts, right? And amines, how do they react? There is essentially one reaction of amines, correct? There is a singular reaction for an amine, which is nitrogen. Nitrogen lone pair will attack something, correct? Nitrogen lone pair will attack something either as a base, but most likely as a nucleophile. Amines are not basic. Amides are, amines are not. Amines are mostly nucleophilic, right? So what type of reactions do amines do? What do nucleophiles do? What's it? What do nucleophiles mostly do? What does a nucleophile mean? Something that likes a positive charge. So a carbon with a positive charge. Hydrogen with a positive charge is called a proton. And if something takes a proton, do you call it a nucleophile anymore? No, you call it a base. So if you are referring something as a nucleophile, what is it most likely reacting with? A carbon. Right? And most likely that carbon will not add because carbon already has 4 bonds. So when will this happen? When it loses a bond. Right? So this carbon can only take the electrons from nitrogen when it loses something else. So is that essentially substitution? And for the most part, it is substitution. So what is important in substitution? Sterics. Right? So bulk on the substrates makes substitution fast, slower. If there is a lot of bulk, the reaction becomes slower. Less bulk means faster reaction. So primary amine versus secondary amine. What has more bulk around the nitrogen? Secondary has more bulk. Secondary to tertiary. What has more bulk? Tertiary. So what should react the slowest under substitution conditions? Right. So does that mean primary should react faster than secondary? Secondary should react faster than tertiary? So under identical conditions, would you expect the secondary amine, primary amine to be the most reactive and hence most potent? Is that what we said? I'm not saying this is universal, but just using chemical logic, you could say that secondary amines are more potent than tertiary amines. Let's say there is a drug. It's potent but not highly potent. There is a tertiary amine in there. What is an easy thing that you can try and test to increase the potency? So there is a drug. It has a tertiary amine. It is less potent. You want to increase its potency, and you're at the structure activity relation stage. Let's say you're making changes. What change do you make first? Convert that tertiary amine to a secondary amine or a primary amine. That is a highly established pathway to increase the potency of a drug. Is this a chemical modification also?
Yeah.
Yes. So, in the body, yeah, let's stop. I will continue next class. Is next class the exam?
Yeah.